recording. Today is the 28th. Okay, focus, Pinky. You have probably heard it in context of like, yeah, I got the gene for curly hair. Oh, yeah, he's got a gene for green eyes. Oh, my dog has a gene for a merled coat. That's how you've heard it? Yeah. What is a gene actually, though? Honestly, I've never heard that. You've never heard that? Is it a trait?
It's a let me pass on.
Something that's passed on.
It's a trait from your aunt.
Well, no, it's not the trait at all. A gene literally has a physical meaning. It is literally a chunk of DNA. That chunk of DNA controls the trait that you're talking about. So it's not the trait. The trait is curly hair versus straight hair. The trait is cowlick versus no cowlick. The trait is brown eyes versus blue eyes versus green eyes. The trait is crooked pinkies or not crooked pinkies. The gene is a section of that DNA. So we've got this mess here, and this piece might be a gene. Now, there are miles and miles and miles of DNA in each of your cells. We said that. And I believe, I always forget this number, and I always need to look it up in advance, and I always forget to. Who's my web fact checker in here? Who's got flying fingers? I want to say there are about 30,000 known genes in the human genome. Somebody can fact check that if they want to pop how many genes the humans have. Thank you, Ian. An individual gene contains instructions for a couple of proteins. Some genes, sometimes one gene equals one protein. Sometimes one gene equals a few proteins. You should probably write this down. That is brilliant. There are some, how many genes? 100,000? Why did I think there was a 3 in front of that?
300,000.
Okay, now I'll take your word for it. Okay, so I could have sworn there was a 3. But okay, we'll say 100,000 genes in the human genome. So genes can code for 1 protein, 2, 3, 4, 5. I don't know if there's a maximum on that. I don't know if there's an upper limit. But I know that it varies somewhat. So genes can also be different sizes. Ha! Ha! Get like genes? Genes come in different sizes. I know. Thank you. I don't care if you're mocking me. Some genes are, 10 base pairs long. Some genes are a couple hundred base pairs long. So it all depends on what you're building. It all depends on what you're building. So genes do come in different sizes, and they code for different numbers of proteins. Now, here's where we get into a little difficulty. Where's your DNA? So the question is, where's your DNA? Well, in the bacteria, we can see the DNA very clearly. It's right out there in the middle. There's really no place to hide it. It's like a one-room apartment. Everything's right there in the open. But if we zoom into the cellular level in you, this is my zoom in. Get it? We're looking at an individual cell in your hand. It's not very good, but it's the best I can do. So, here's your cell. Notice you don't have a cell wall. You are a eukaryote, not a prokaryote. Bacteria is a prokaryote. So you don't have a cell wall. You still have some of those little, I shouldn't make those green. Let me change my colors. Okay, so the bacterial cell, like I said, DNA, ribosome, cell membrane, cell wall. Maybe a flagella or two. However, you glorious multicellular organism, you got a little bit more going on. Oh yeah, you have a multi-room apartment. So in that cell of yours, number one, there is no cell wall. There's just a cell membrane. Number two, you've got the ribosomes. You have a nucleus. And you do some cell stuff in middle school, I think, correct? So you know that if you memorize this is what they typically give you at the middle school level. The nucleus is the control center for the cell. Have you heard that? The nucleus is the command center for the cell. The nucleus controls the cells. Well, that's all true, but why does the nucleus do that? Because guess what the nucleus has in it? Oh, zooming in. DNA. That's where you're storing your DNA. So, and I cannot draw the amount of DNA. And you've got other stuff in that cell. You've got things like Endoplasmic Reticulum and Golgi. I like saying Endoplasmic Reticulum. It's fun to say. And you've got Golgi. You've got all kinds of other stuff in that cell. But the important thing is you have a nucleus that's containing your DNA. You have a nucleus that is containing your DNA. Don't open that. Don't you dare open it. Don't you dare. If you open it, I will probably stab you with a pen.
That would hurt.
That would hurt. You can shake it. I don't care if you shake it. You can't open it. So inside each cell in your body,
I tightened it.
Your body and the body of a maple tree and an earthworm and a blue whale and, let's see, a ladybug and a dragonfly and a dandelion and a mouse. Yeah, that's okay. You don't have to be fooled. I'm not trying to fool you. Would I try to fool you?
Yeah.
Have we met? Would I do something like that to you? Yeah, well, maybe. Okay. So, inside all those eukaryote cells, there's a nucleus, and inside that nucleus is that DNA. Now, here's the question. Sorry. If I've got this big, long piece of DNA, and I want to build the proteins associated with just this little section, do I have any problems to contend with? So I've got this giant mess of DNA, and it is messy. Sloppy, very, very sloppy. And what I want, yeah, I know, is this little teeny tiny section right here. I just want to build the protein associated with that. So, I mean, this is first off just a royal pain in the butt. Because how do I get just that one section of DNA? This is like, you know, a 3-volume encyclopedia cookbook, and I need a recipe this big on page 700, and there are 3,000 pages. I don't really want to drag the whole cookbook out. I would much, much rather be able to just copy that page down onto some note card and carry it with me. So that's my first problem, is I've got a giant mess that I have to sort through to get to the recipe I want, to the gene that I want to make the protein that I need. Now, bacteria, as you might imagine, have it a little bit easier. But even on a piece of bacterial DNA, they're still coding for multiple proteins. There are still multiple recipes there for multiple proteins, and we only need a portion of it. So we might only need this. We don't need the whole thing. So that's the first issue is, you know, in either case, we just need a snippet of what's there. But here's the bigger issue. So you can look at that thing I put on your desks, and you'll notice that there's a hole. Find the hole. So that nucleus is actually covered in holes. I only put one hole in your, and this is, you figured it out. It's a cheap, shabby model of the nucleus. That's what you get for, that's what you get. That's what you get here anyway. I don't know if everybody gives you cheap, shabby models of the nucleus, but I do. Do you think that you could successfully extract the entire length of DNA that's in that cup out through that tiny hole without tangling it or breaking it?
Probably not.
Probably not. Do you think you would be able to do so in a way that was efficient and you could just get the section you wanted to read? Seems sketchy to me. So here's the thing. That DNA never leaves the nucleus. So because of that, because it would just be a hassle in you, or a maple tree, or a blue whale, or a tulip, or this peace lily, or your dog, or an earthworm, or a firefly, or dragonfly or a ladybug or a damselfly, or I like bugs. A caddisfly, ooh, an alderfly, ooh, yeah. A butterfly, a moth, a skipper. Any of these eukaryotes, DNA never leaves the nucleus. Do you remember where proteins are built? Where? Ribosomes. Ribosomes, little protein factories. Illustrated here as blue dots. So they're outside the nucleus. So how do we build proteins if the instructions are locked in a box? So in order to get the instructions out of the lockbox, we can't take the whole book out, but we can copy down a segment of that information and we can move it outside the nucleus. And the copy we make is smaller, it's more compact, and it only copies one gene at a time or two genes at a time. And of course, most of you said the copy is RNA. So the copy is half of the DNA. Now, in bacteria, and we're going to compare and contrast you and bacteria a lot, because of course we're really interested in protein synthesis in bacteria, because we want to know how it relates to what? What are we talking about this whole year so far?
Bacteria.
Antibiotic resistance. So we need to know this because a lot of antibiotics work by stopping bacteria from making protein. They actually interfere with their bacterial protein synthesis. That's how they kill them. They don't stick pins in them. They don't dry them out. They don't burst the cell wall. Some of them do, but a lot of them work on protein synthesis. Actually, later in the year, probably December, January, we're going to start talking about Lyme disease. Anybody here know someone who's had Lyme? Me too. Lyme disease, the antibiotic of choice for Lyme is doxycycline. I know somebody who was told that to actually get better, he would be on doxy for about 15 years. Yeah, he had had late-stage undiagnosed Lyme. Yeah, and doxy does a number on your stomach. It does a number on your teeth. It makes you sunburn easily. It's miserable stuff. Doxycycline interrupts protein synthesis in the Lyme spirochete. So that's why we want to know this. So we're going to compare and contrast those. Now, in bacteria, there's no nucleus to leave, but we still make an RNA copy because you only need a little chunk of the DNA. You don't need the whole thing. All right. We've already done one thing with your DNA, your metaphorical DNA. We replicated it. Everybody have your DNA?
Yeah.
Get out your DNA.
My DNA?
Get out your pipe cleaner DNA. Get it out. Get it out. Get it out. What was the first step when we wanted to replicate DNA? What did we have to do to it?
Unzip it.
Unzip it. Guess what we're going to have to do to this if we want to make a copy of half of it? Unzip it. We're going to have to unzip it to make a half a copy. Okay, so you have to unzip it. You have to unzip the DNA, and then we are going to do something weird. Are you excited about that?
Yeah.
No?
I like weird.
I tend to kind of be a fan of weird. So I need to pick a new color. Okay. I'm going to make an RNA copy of half of this. And RNA is a little different. We only make one side. Single-sided copy. So we need a smaller molecule to get out of those nuclear pores. And there's one other difference. So G pairs with what?
C.
C. So G pairs with C. C pairs with G. T pairs with A. A.
Hey.
Oh, what happened there? I squished something. I had a mutation. There's a U. Every place that you would have inserted a thiamine, there's a chemical called uracil. Uracil. Yeah, Yeah, it's one of your vocab words. You've seen this word before. Uracil is the base that exists in RNA that doesn't exist in DNA. And every place that you would normally put a T, you put a U. So A pairs with what?
You.
U. A pairs with U. And U, and U, and U.
And you.
And U. Exactly. What does T pair with? Not a trick question. A. A. No, sweetie, not a trick question. What does A pair with? U. Okay. So that is a completed piece of RNA. Now, step 2, in you, or a horse, or a coyote, or a maple tree, or an earthworm, what does the RNA have to do to go out to the ribosome? This sounds like an adventure action flick. Escape from the nucleus.
Oh.
Dun,
Dun dun! Oh, you know what? You could make a iMovie trailer for that. That'd be great, actually. Wouldn't it? They thought it couldn't be done. They thought no information could be taken out. Little did they know. Dun dun dun! In theaters soon. Escape from the nucleus. So it actually, the RNA actually leaves via those little pores. So those are called nuclear pores. So in you, the RNA copy, this is the RNA, has to escape from the nucleus and then travel to the ribosome. In bacteria, it just heads straight to the ribosome. There's no escape necessary because there's no nucleus. Quit playing with your nucleus.
coming out.
Well, the DNA never comes out. The DNA can't leave the nucleus.
Falling out.
Once it travels to the ribosome, there's a multi-step process, and that's what we're going to start on Monday, because we're going to have some props. So I did ask you to get out your helical DNA. We're going to actually generate the RNA with that today. Okay, so let's look at this for a second. I took a picture of one of yours. This is my DNA. I have 2 strands. Okay, so focus. Focus. We unzipped the DNA. We made the RNA copy. The RNA traveled out to the ribosome. Then the DNA zips itself back up. The DNA just zips itself back up. It keeps going about its business. Perfect. Maybe another section of it unzips and makes another piece of RNA. But once the RNA's been made, that DNA can zip back up. Okay, so we've got RNA unzips, DNA unzips, make an RNA copy. RNA travels out to the ribosome. DNA rezips. DNA is done. DNA's part here is done. The DNA has nothing else to do with protein making. It has the instructions. The instructions have been copied. On Monday, we're going to take those instructions and we're going to make some proteins. Now, if you compare, so look at the picture for a second. Look up your screen. Your RNA strand should look an awful lot like 1/2 of your DNA strand, except for the uracil. So this RNA was made from this half of the DNA. No, it was made from the brown half. Look. Because it almost perfectly matches the green half. Because it's complementary bases. Okay, that's where we'll stop, and Monday we make protein.